So you've heard a lot about Florida already today, so I apologize, bear with me. And I think I can move pretty quickly through some of this because a lot of it's redundant from what Dave shared with us and even a little bit about what we just heard. So I'm coming to you so I work for Florida Fish and Wildlife, and we do the biotoxin testing for the state. We also do HAB monitoring for the state. And so I work, our program works within the NSSP, the National Shellfish Sanitation Program. But we've also had a few success stories where we've been able to participate in the process with the ISSC and actually make changes to the program that have benefited us and can benefit you as well. We've seen this chart already. These are the toxins for which there are U.S. regulatory limits, and so they're in the NSSP. There are organisms in the Gulf that produce every single one of these. So even though we're mostly focused on Carenia, they're all a potential problem. In Florida, we have very focused monitoring programs, and they're different for PSP, NSP, and ASP. The model ordinance is pretty general in the guidelines about what your biotoxin monitoring should be. You just have to meet these guidelines. You need to have one in areas where these organisms are known to occur. It can be tailored to those times of year when the organisms are known to be there. And it's based on sampling water and or shellfish for the cells and the toxins in the meats. In Florida, we do this through a partnership. So I'm with FWC on the right, and we do the HAB monitoring and the biotoxin testing. We are not the Shellfish Management Authority. That's the Florida Department of Agriculture and Consumer Services. They classify the waters. They go out and they sample the shellfish harvest areas. We do a lot more sampling outside of those areas as well as near those areas. And together, the biotoxin plan comes together. We were at one time under the same roof in the same agency. A bunch of reorganizations happened over many years. Well, I think at the time it was the Florida Department of Natural Resources. Everything got split up, but this separation of responsibilities really works well. And having the harmful algal bloom group within the Fish and Wildlife Agency is especially helpful with red tide because of the devastating impacts it has on fish and wildlife. So we work very closely together to respond to fish kills and marine mammal mortalities, as well as doing the shellfish side of things. But on paper it's two agencies. In reality, it's a village. We really rely on many partners, and that's where I think you guys can start to look to get some help. We work with all the other state agencies, some of the federal agencies. We work with local government entities who help us collect samples, and we have a pretty big army of citizen volunteers too, who go out and collect big samples for us, places we could never get to, offshore fishermen, for example. And here's an example of the difference it makes. So on the left are the samples that FDEX collected in shellfish harvest areas in the month of October last year. On the right are all the samples we got in the month of October last year. So in all, there's 2,000 samples up there. Given that was an extraordinary month in the middle of an extraordinary event, but it really does make a huge difference because these blooms, they do move around. They can be there one day, not the next. And you can see if you just stick to the shellfish harvest areas, you don't really get the full picture. So in a nutshell, how we monitor NSP, FDEX will collect samples weekly, and it's only weekly. It's really hard to go beyond that because there's so many other things they have to do. They have maybe 5 field offices around the state with 2 people in each one that have to do all this too. So they collect samples weekly. They send them to us weekly from Southwest Florida. Now there's an example of sort of geographically localized. That's where our blooms manifest initially. So that's where most of the focus is on for Carenia monitoring. They'll collect monthly in other areas around the state. And any samples that have greater than 5,000 cells per liter will initiate a closure of the zone, of the shellfish harvest zone. As you've heard referred to, they've started to kind of micromanage some of the areas. The ones that have high density lease areas, they've designated authorized use zones, and those are for commercial clam farmers. That's mostly what we have in Southwest Florida. So while the greater shellfish harvest area is closed for recreational harvest of any species, the authorized use zones remain open as long as the cells are not there. Once they are there, they'll have to close too, but then we have another program that I'll talk about too that can help. And once the counts go below 5,000 cells per liter, and meat samples are less than 20 mass units or below the approved ELISA threshold, which is different. It's not a new action limit. It's a very conservatively set limit. And in oysters, it's 1.8 ppm, and clams, it's 1.6 ppm. So if they're below that, we can reopen. If they're not below that, we can do a mouse bioassay. If they pass the mouse bioassay, we can reopen. We talked a little bit about precautionary closures today too. Some of the things that will initiate a precautionary closure in Florida is if we have counts that are over 50,000 cells per liter in an area that's near a shellfish harvest area, until they get more information. Or if my agency goes out and collects a sample in a shellfish harvest area, or one of our partners does, if it's over 5,000 cells per liter, they'll issue a precautionary closure, but they'll only do an actual closure on samples that FDAC has collected at their indicator stations themselves. They'll also do it if we have early warnings, such as fish kills in the area or reports of respiratory irritation. So they'll be conservative and do a temporary closure, but then if they go out and sample and there are less than 5,000 cells per liter, they'll go ahead and reopen. They don't have to grab a shellfish sample, but they probably will. And this is just one example of a pretty important area that's routinely impacted by red tide in Southwest Florida, Pine Island Sound. So you can see the green hatches are approved harvest areas, and then the 12 yellow pins are sort of their routine sampling sites, just to give you a sense of distribution within a shellfish harvest area. We do monitor for PSP. We have had multiple closures for PSP in shellfish above the regulatory limit. We have never had a human illness from PSP in Florida. Most of our problems are where that red circle is on the East Coast in the Indian River Lagoon. And so our monitoring program is geographically restricted to that area. You've seen where we collect water samples. Every water sample we get is enumerated for Crania, for Pyridinium bahamense, which is our PSP producing organism, and for Pseudonychia. So they're covered in all the samples that we look at. But specifically targeting PSP monitoring, we'll get monthly water samples from that area, and then come April, because it's not perennial, we don't get the early warning, we don't get the fish kills, it's not coming from offshore, it lives in that system. They'll start sampling meats in water monthly in April. It's a summertime thing, so they get a few months ahead of it. And we'll look at the water and we'll screen the meats with the test that Dave showed up there, the Scotia Rapid test. Those trip pretty early. As soon as they get a positive on those tests, they'll close the area and we'll do a mouse bioassay. If the mouse bioassay passes, they'll reopen, but then they will increase the frequency of where they're sampling, and we'll do routine meat testing, and we'll do more frequent water sampling, and they'll also expand the area because it can go north and south within that system pretty quickly. Let's see. Well, that's about it. For it to reopen, unlike NSP, it has to pass a test twice, at least a week apart. So because of the lethality that could be associated with that bloom, it's kind of take it a little bit more seriously, and we have to have 2 passing tests at least a week apart. We've heard a little bit about Pseudonychia, which is a diatom that can produce domoic acid. We do an awful lot of testing for that toxin in all those water samples that we get in. Any water sample that comes through the door that has more than 100,000 cells per liter and is not preserved, we will test for domoic acid. So we have a wealth of information. The bracketed areas are areas where there's shellfish harvest and where we have like a high number of samples. And about half the time, we'll measure domoic acid in those samples where there's greater than 100,000 cells per liter. So it's not always toxic. We've noted it can be environmentally induced, but it can also be, very often we think it's species succession. You can't really tell the species apart, but we'll do genetic testing on all our bloom samples, and we can watch the bloom shift from one species assemblage to another. Commonly, we have pseudonychia in the water, but the toxin concentrations are typically on the low end, and you can see where that median line in these box and whisker plots is, you know, a microgram per liter or less. I would say a microgram per liter is, you're getting high. I pay attention, I start talking to FDACs, but the only closures we've had have been in St. Joe Bay when we've had like tens of millions of cells per liter and about 10 micrograms per liter of domoic acid. So our plan is not specific to any one area, even though, and it's not specific to any time at all. It's statewide, and kind of mentioned this already. They collect their samples monthly, we'll test them for domoic acid. Once they get up to 1 million cells per liter, which is the same threshold you guys have here, in St. Joe Bay, they would instantly do a precautionary closure. Anywhere else, they will try to run out and get meats. And we'll try to test the water as soon as we can, like within a day or two. If they can't get out there, if it's a Thursday, if we're not going to be able to test the meats or the water anytime soon, they'll do a precautionary closure. And they've done multiple in different areas. Areas will only be closed if the shellfish domoic acid levels reach or exceed 2 milligrams per 100 grams. So it's a pretty, compared to other toxins, it's a pretty high threshold. And as we said, only St. Joe Bay. And there have been 3 events. One of them happened to coincide with the start of scallop season, and that was the biggest disaster we've had up there. Okay, so I'm going to segue briefly and talk about methods. I'm not going to go through the table Dave mentioned. There's a million different ways to look for these toxins. You know, there's scientists all over the world, but there's only a few approved ways to test for these toxins, and that's the problem. PSP has the most options. For NSP, the only approved method is still, the only fully approved method is still the NSP Mouse Bioassay. Approved limited use methods, primarily they're used for screenings and precautionary closures. The Abraxas shipboard ELISA, as Dave mentioned, is used to determine whether or not you can even harvest. But then that product is tested with an approved method. But the Marbionic ELISA, and I have put the company in there because there's a few different Biotoxin ELISAs out there. That's the one. They're all different. That's the one that's approved limited use. You can actually reopen shellfish harvest areas with that method. What makes it limited is the species you can use it on and the fact that it has this threshold you have to stay below. The NSP Mouse Bioassay are approved methods right now. There are 2 labs in the state, in the country that do it. There's a lab in Texas and there's my lab. And just to give you a little sense of what it's like and why it's not ideal, I like to show the process. So the authority does the collections. They even shuck up the samples for us, which is awesome. And they'll drain them, and it's a big state. So more often than not, they're shipped overnight. They're not delivered the day that they're harvested. So we get the samples on day 2. On day 2, we homogenize them. There's a little cooking step with acid. And then there's a manual diethyl ether extraction, and it's pretty high volumes of ether. And it's explosive. It's dangerous. You can't do that automatically with a machine. So we have people literally shaking bottles in a spark-proof lab. And you have to do each bottle 4 times. And at the end of it, you have multiple beakers with large volumes of ether that just have to evaporate by themselves overnight. You can't stick that in a rotowrap. And so like day 2 is all extractions. And if on a normal day, we'll do 6 samples max. And that takes 2 people. And then on day 3, you have to put that lipid residue into a vegetable oil. It's pretty gross. And so you have a 100-gram extract that goes into a 10-gram equivalent of oil. So it's really nasty stuff. And each mouse gets injected with a mil of that. So one mouse is getting a 10-gram equivalent injection into its peritoneal cavity. We have to do 5 mice per sample. This is not like PSP. It's not a quick assay where you can do these 1 mil aqueous injections very rapidly and watch the mice for 5 minutes. Every single injection is slow when the mouse is moving, and you have to start your clock and watch what it's doing. So it can take a long time just to get them going, and you have to watch them for 6 hours. It's not a high throughput assay at all. So if you're lucky, by 5 o'clock on day 3, you have an answer. The good news is that it has successfully prevented any NSP illnesses from legally harvested shellfish since it was implemented. Some will argue that it's, it's a conservative level, this 20 mouse units. It's really not based on a toxicological study. It's the lowest level that you can reasonably detect, given that a lab could reasonably detect within a day, like between getting the sample ready and doing a 6-hour observation. That's your limit. At 20 mouse units. 20 mouse units is the amount of toxin that it will take to kill half the mice in 6 hours. It's a weird thing. On the con side, it's low throughput, very labor costly. It's non-specific, and we do have problems with that, false positives. It's not calibrated against any known concentration of toxin, and of course it uses live animal testing. So we need better methods. We haven't had any because Biotoxin is a really complicated family of toxins. There hasn't been any agreement on what we should be looking for. There are no analytical standards. So there's that problem. Until sort of recently, there was no clear path to getting a method approved for the ISSC. And then compared to the other toxins, this is a fairly localized issue. It affects us, it affects Texas. It's starting to affect you guys more, but nobody else cares about it. It's just us. But we've kind of made some progress. So there's an ELISA method that I mentioned. ELISAs are a general type of a assay. You can use it to measure almost anything. It uses antibodies that are raised in animals, goats in our case, that recognize the antigens, which are the Biotoxins. Lots of different formats. It is just a screening assay, and it gives you an overall estimate of what's in the sample. This is just some idea of how long it's taken us to get from A to B. The antibodies were developed in '91. The assay was developed in 2001 at FWC's request. 8 years of research during which we began to understand the metabolism of Biotoxins. And then we got funded by FLORIDA grant in 2014 to do validation using the ISSC requirements that were finally out there and available to use. And we got our method approved for limited use in 2017, and now we're in the implementation phase. So I already talked about that. This is how it's limited use. This is how it's allowed to be used. If it's below those thresholds, you're good to go. At or below, I'll say. If it measures 1.8, you're good to go. If it's above that threshold, doesn't mean it fails. You can still do a mouse bioassay and reopen your area. Just you can't take any management action based on a failing ELISA. And we have started transferring this technology to the other states. We were here in April in Dr. Robertson's lab with the Alabama Department of Public Health folks, and you might recognize Drew and Lonnie and Aaron there. And we have workshops planned with Mississippi next month, and we have workshop planned in Texas in August. And that's funded by NOAA. And I'm out of time. We already talked about this, controlled harvest of shellfish with end product testing. This is an example of how it would work. Within that red polygon, that's the quarantine zone. So if the whole area is closed for Corynea, as long as cell counts within that red area are below 50,000 cells per liter, then the farmers can opt to do the quarantine, and they can harvest some product. We'll test duplicate batches of that product, and if they both pass, it's good to go. If either lot fails, the product has to be destroyed or returned to the lease. And I'll tell you though, since they started doing that micromanaging of the shellfish harvest areas, by the time that polygon closes, they need to be closed. Like quarantine works better when they close the whole shellfish harvest area, and we did that. Now that they wait until the bloom is really on top of them, they need to be closed. They fail. So they're not happy about that, but they stay open longer. And that's just the language for what's there. So